Hi, in this video I will be looking at A2 Biology of the Edexcel exam board, in particular Unit 4 Topic 5, which looks at ecology, evolution, natural selection, speciation and the succession of species. Ecology is the study of interactions among organisms and their environment. An ecosystem is defined as all organisms living in a particular area and all non-living, so abiotic factors, which we will talk about shortly. So how does energy transfer through an ecosystem? So the main route of entry of energy is through photosynthesis, where sunlight is converted into chemical energy by plants, which then can be used by other organisms. So plants are the producers, and primary consumers consume grass, they can get some of that chemical energy. And energy is transferred from producers to other organisms when they're eaten. Primary consumers are then eaten by secondary, and the secondary consumers are eaten by tertiary consumers, as shown here. Primary, secondary, and tertiary, and this is in a form of a pyramid. And at each stage, energy is lost. So producers represent a trophic level, primary is also a trophic level, so then we can later on see how we can calculate the energy transfer between the different trophic levels. So we can represent the energy transfer either with a food chain, as shown here. So it's a simple diagram of lines showing how energy is transferred. So we have energy from the sun, is then changed into chemical energy by producers through photosynthesis. Then when the grasshopper eats the grass, he then gets some of that energy, and so on. Or we can use more complicated diagrams, which are known as food webs. In terms of energy transfer, not all of the energy available to an organism in one trophic level is transferred to the next level. About 90% of the total available energy is actually lost through metabolic processes. So out of the 90%, 60% of it is not taken in, because for example, not all the light is used in photosynthesis, because some of it is the wrong wavelength, or it's reflected, or it hits the bark of the tree, where photosynthesis can't take place. Also, some parts are not eaten, so for example, roots and bones. And also, there may be indigestible food, which is then passed through the organism and is excreted in the form of feces. The remaining 40%, which is absorbed, is known as gross productivity. But again, not all of it is available for the next trophic level. 50% of the gross productivity is lost to the environment, and this is known as respiration loss. So the energy used to move or to maintain body heat of an organism. And the remaining 10% goes into biomass, which is stored or used for growth, and this is known as net productivity. So here I've got a diagram where we can see we start off with 1,000,000 joules of sunlight. For the primary producer, only 10,000 of those joules are left. For the primary consumer, only 1,000, secondary only 100, and then the tertiary only 10 joules. So it's only about 10% efficient. So let's look at some of the calculations that we need to know how to do. So net productivity is the amount of energy available to the next trophic level. So to get the net productivity, we need to minus respiration loss from gross productivity. We can also be asked to calculate the percentage efficiency of energy transfer, in which case we take the net productivity of a level divided by the net productivity of the previous level times 100. Net primary productivity and gross primary productivity applies to producers. So to get the net primary productivity, we need to minus plant respiration from gross primary productivity. So I thought we would look at a question, because it's easier to understand when we go through a question. So here we're given the numbers. So from grass we get 13,883 kilojoules. When the Arctic hare eats the grass, it gets 2,345, and when the hare is eaten by the fox, it gets 137. So question A, explain why the net productivity of the hare is less than the net productivity of the grass. So this is because not all of the energy is available from the grass is taken up by the Arctic hare. Some parts of the grass aren't eaten, so the energy isn't taken in, and some parts of the grass are indigestible, so they pass through the air and come out as waste. Some energy is lost to the environment when it's used when it uses energy from respiration for things like movement and body heat. And B is asking us to calculate the percentage efficiency of energy transfer from the hare to the fox. So the last two stages here. So the efficiency of energy transfer, how do we calculate that? So we take the net productivity of the level, which is the last level, which is 137. We divide that by the net productivity of the previous level, which is 2,345, and then we multiply that by 100, and we get 5.8%. So the efficiency of energy transfer is 5.8. So let's also look at some definitions that we need to be familiar with before we go into more detail. So habitat refers to the place where an organism lives. So a field, you can simply remember it as the address, if it helps you to remember it. Population is defined as all organisms of one species in a particular habitat. Population size looks at the number of individuals of one species in a particular place. And abundance is also the number of individuals of one species in a particular area. Population size. So is population size and abundance basically means the same thing. So you can refer to it as either population size or abundance. Community is the population of different species in a habitat that make up a community. Abiotic factors are non-living features of an ecosystem and include things like temperature, availability of water, light conditions, soil, the pH, humidity, the content of the air, wind, and other inorganic nutrients, such as nitrogen or phosphate. Biotic factors are living features of an ecosystem, so these include predators and food. And finally, distribution is where a species is found within a particular area. So for example, here we have a map of America, so we can see the dots represent the distribution of a particular species throughout North America. So let's look at abiotic and biotic factors and how these influence the abundance, so how both abiotic and biotic factors vary the population size. So let's first look at abiotic factors. These include light, water and space availability, and the temperature of the surroundings, or also the chemical composition. So when we have ideal conditions, and let's say we have an ideal temperature, so metabolic reactions don't need to use up as much energy to maintain body temperature. Therefore, more energy is available for growth and reproduction, so the population size will increase. If we deviate from the ideal conditions, so for example, when the temperature is lower or higher than the optimum, the organism will need to use more energy to maintain the right body temperature, which means that less energy will be available for growth and reproduction. Therefore, the population size will decrease. In biotic factors, there are three main things we need to know about, which is an interspecific competition, intra-specific competition, and predation. So interspecific competition refers to the competition between different species for the same resource. So for example, in the UK, we have red and grey squirrels, which are part of different species, but they're competing for the same resource. The available resources are reduced for both species, which means that both have less energy for growth and reproduction, so the population size will decrease for both species. Intra-specific competition refers to the competition between the same species for the same resource. So the population size will increase when there is a lot of resources available. However, after some time, the space becomes a limiting factor. So the population will decline, which means that if there is a smaller population, there is less competition, meaning there is more growth and reproduction, which then leads ultimately to, again, an increase in the population size. And predation is when we have a predator which kills and eats the prey. The relationship between predators and prey is interlinked, however it's very complex, so I'm only mentioning the sort of the basic idea. So as the population of the prey is increased, this also means that the predator population also increases because there is more food available. But as over time the predator population increases, more prey is eaten. So eventually the prey population starts to decline, which then leads on to less food being available for the predators, therefore a decline in predator population will happen. So let's look at how abiotic and biotic factors affect distribution. So abiotic factors, for example, some plants grow on the south-facing slopes in the northern hemisphere as the solar input is greatest there. However, some plants don't grow near shorelines because the soil is too saline. And also large trees can't grow in polar regions because the temperatures are too low. So we can see this is how abiotic factors, so non-living factors, affect where we find certain species of animals or plants. Biotic factors, for example, interspecific competition, affects distribution, where one of the species may be better adapted than the other, so the less adapted species won't be able to outcompete, which means that they cannot coexist. So, for example, grey squirrels, which were introduced into the UK, have now completely replaced red squirrels because grey squirrels are fatter, so they can survive winter. And also I have a picture here which shows the distribution and the abundance of the red kangaroo in Australia. So we can see that with the more intense color shows where the abundance of kangaroos is higher. The conditions are semi-arid or arid, which means there is less precipitation and relatively low variability from year to year. Whereas if we look on the outside areas, so for example the climate in northern Australia is very hot and wet with a seasonal drought, so there are no kangaroos there. So this relates how biotic and abiotic factors sort of contribute to where particular species can be found. So let's look at niches. So a niche is defined as the role of a species within its habitat, and it's the sum of a species' use of biotic and abiotic resources and its environment. An easy way to remember it, if you remember that an organism's habitat is its address, and the niche is its profession. So biotic interactions include what an organism eats and who eats it, and abiotic interactions can include the oxygen that it takes in and the carbon dioxide that it breathes out. So generally, two species cannot coexist permanently in a community if their niches are identical, but they can coexist if one or more significant differences can be found in the niches, which can arise over time. So for example, here we have an example of resource partitioning among Dominican Republic lizards. So because they live in very close proximity and they all feed on insects, but because the competition for food is reduced because each lizard species has a different preferred perch, thus occupying a distinct niche. So this type prefers to live on sunny surfaces, whereas this one prefers to live on shady branches. So evolution by natural selection means that one species is now using a different set of resources, which I've just talked about, which is known as resource partitioning. We also have to know how to carry out a study of a habitat practically. So we can investigate populations of organisms, so we can look at the abundance, so the number of individuals of one species in a particular area, and we can also look at the distribution, so where a particular species within the area we're investigating is found. So there are different ways of doing it. So first we can do a random sample of the area, and this is the shortcut method to investigate the whole population. So first we need to choose an area to sample. So it's a small area within the larger area. We have to use a random method to avoid bias. So we divide the field into a grid, and then we use a random number generator to select coordinates. The area that we are wishing to investigate is this. So instead of grouping all of our samples in one particular area, when we use a random number generator, we can see how they're all randomly distributed. So this creates a better view of the whole area. We then use the appropriate sampling technique, which I'll go through in a minute. We also need to make sure that we repeat the experiment, which gives us more reliable estimate of the whole area. And we can also estimate the number of individuals in the whole area by taking the average from each sample and by multiplying it by the whole area size. So this gives us the percentage cover. Let's look at sampling techniques. So we could use something known as a frame quadrant. So a frame quadrant is a square frame which is divided into a grid of 100 smaller squares, which is shown here on the left. We usually place this on an area of investigation after random assignment of coordinates, and the number of individuals of each species is recorded in each quadrant. We can then calculate the percentage cover of plant species, so how much of the quadrant is covered in a particular plant. And this is quicker because we don't have to count individual plants. We can also use point quadrants, as shown here on the left. It's a horizontal bar. It has two legs with holes along the top which are at set intervals. And we can again place this at random points within the area of investigation. We then drop pins, as shown here, through the top, which has the holes, and then every plant that the pin touches is recorded. And then this allows us to calculate the number of individual of each species in each quadrant. Again, we can then calculate the percentage cover, and this is useful for when you have very dense vegetation or is close to the ground. So shown here, you can see that the grass is quite high, so this is quite a good way of sampling. Finally, we can also use some transect, and this is where we use lines to find how plants are distributed across an area. So we can either use a line transect, belt, so the data collected along the transect with the use of frame quadrants next to each other, or they can be interrupted as shown here. So measuring out intervals instead of the whole transect by placing point quadrants at right angles to each other, let's say every 2 meters as shown here. So they're not touching, there is an equal distance between them. In our practical investigation, we need to make sure that we also measure biotic factors, because these affect the abundance and the distribution of organisms. So let's first look at climate, so weather of the region. So how can we measure that? Well, we can use a thermometer to measure the temperature. We can use a rain gauge to measure rainfall, and a rain gauge is a funnel which has a measuring cylinder which collects the rainwater, and the volume collected over time can be used as an average of the rainfall. We can also measure humidity, so the water vapor content in the air, using an electronic hygrometer. If we want to measure O2 availability, so how much oxygen is dissolved in water, we can use an oxygen sensor, but this only applies to aquatic habitats. We can use a light sensor to measure solar input, so the intensity of the light. We can also look at the content of the soil, so we can use a pH meter, either as liquid, so when it changes color, or we can use one that's electronic. We can also determine the moisture content, and this is done by taking a sample of the soil. It is then dried out in an oven and weighed before and after, and the percentage difference is calculated, which tells us about the moisture content. We also need to describe the shape and the features of the surface, so how the height across an area, so we can use a GPS to see the difference between points. You can also use a map or contour lines. If we have a slope, we need to make sure that we know the angle, so how steep it is, with the clinometer. A simple clinometer would include a string with a weight attached to a protractor, which gives us a reading of the angle. Also, we need to make sure we know the aspect, so the direction of the slope, either north, south, east, or west, and we just use a compass for that. So let's look at ecological succession. Succession is the establishment of a community in an area of newly formed land, so for example, new volcanic island. And secondary succession refers to the transition in species composition of a community following a disturbance, so for example, a forest fire or deforestation. But in secondary succession, the soil is left intact. So here on the left, I have a picture of a forest fire in Indonesia, which means that this will be a secondary succession. And here we have a pioneering plant after the fire. So there are stages of succession that we need to know about. So the primary stage is when species colonize a new land surface, and the first species are known as the pioneer species. So first of all, the abiotic conditions are very harsh, so only pioneer species can grow because they're very specialized. So for example, marang grass has very long roots, so it can get water and can tolerate a high salt environment. The pioneer species change the abiotic conditions as they die. Microorganisms decompose the dead organic material, so the soil becomes more basic, which now means that the conditions are less hostile as the soil becomes richer and it can retain more water, which allows larger plants and shrubs to grow. So this leads on to the secondary succession, which allows the plants and shrubs to grow. At each stage, animals and plants that are better adapted move in and outcompete the existing species, so the new species becomes dominant. Over time, the ecosystem becomes very complex. New species move in alongside existing ones, therefore increasing the species diversity and the abundance. And the final stage is where we produce a climax community. And a climax community is defined as a community being able to support the maximum number of plants and animals which remain stable and won't change for a long period of time unless there is a disturbance. So here I've got a picture of glacier retreat and primary succession at Glacier Bay, Alaska. So we can see here the pioneer stage, where the firewood is dominant. After some time, the conditions are changed, the soil becomes less hostile, which leads to a new type of shrub growing. Later on, after some years have passed, trees can begin to grow, and we can see the fourth stage. And these pictures have been taken about 40 years apart, so it takes quite a long time for it to go from the pioneer stage to the spruce stage. So let's look at ecosystem and climax communities. So originally scientists believed that biological communities were at an equilibrium, so at a balance, unless they were seriously disturbed by human activity or another event. However, the difference in the soil, the topography, and other factors create many potential communities that are stable within a region. So, for example, a climax community for a particular climate is called climatic climax. So, for example, in a temperate climate where there is large stores of water and the temperature is mild and the seasonal changes are not very drastic, you will have many large trees because they can grow and develop. Whereas in a polar climate, there is not much water, extremely low temperatures, and there are massive changes in the seasons, large trees can't grow, so the climatic climax contains only herbs, shrubs, but it is still referred to as a climax community. We can prevent succession. So, for example, agricultural development disrupts ecological succession artificially. So the climax community is referred to as plagioclimax. So, for example, logging, clearing for urban development, mining, and farming has reduced large forests to small patches of disconnected woodlots. So I have a number of pictures here. We can see these are satellite images. So this red one at the top was taken in 1992. We can see that there is a river and a very dense forest. However, in 2006, we can see that the forest has been reduced to sort of patches, and in between we have a lot of farmland. The same is shown here. The same goes for tropical rainforests. They're disappearing because of clear cutting for lumber, cattle grazing, and farmland. So this is how you can prevent succession through human intervention. So let's look at evolution and natural selection. So evolution refers to the change in inherited characteristics of biological populations over successive generations. So this is the change in allele frequency, which gives the rise to variation. Gene pool refers to the complete range of alleles in a population, and new alleles are generated by mutations in genes. So the changes in the base sequence of DNA happen during DNA replication, which lead to mutations. Allele frequency refers to how often an allele occurs in a population. So as when frequency of an allele in a population changes over time, this is what we refer to as evolution. Evolution happens by natural selection. So individuals in a population will vary because they have a different set of alleles. Some of those individuals will be better adapted to the environment. So if they have the beneficial allele, which increases their chance of survival, which means they're more likely to reproduce and more likely to pass on the allele to the offspring, which means that a greater proportion of the next generation will inherit this beneficial gene. They in turn are more likely to survive, reproduce, and pass on this gene. So the frequency of beneficial allele will increase with every generation. Let's look at speciation, and this is the development of a new species. And a species is a group of similar organisms that can reproduce to give fertile offspring. So when populations of the same species become reproductively isolated, this means that there are changes in the allele frequencies, which can cause changes to the phenotype, so they can no longer breed to produce fertile offspring. There are different ways by which reproductive isolation can happen. So the things that we need to know about are seasonal changes, mechanical changes, behavioral changes, geographical isolation, and random mutations. So seasonal changes, things like different flowering and mating seasons. Therefore, individuals of the same species may become sexually active at different times of the year. Mechanical changes refers to the changes in the genitalia, which can prevent successful conception of offspring. Behavioral changes can refer to one group of the same species developing rituals, courtship rituals, that are not attractive to the main population, which prevents them from breeding. Geographical isolation, which I'll talk about in a minute, and random mutations can also happen and prevent a species from breeding. So geographical isolation refers to when there is a physical barrier which divides the population of a species. For example, floods, volcanic eruptions, or earthquakes. So here I've got a picture at the bottom. Two types of squirrels that were divided in the Grand Canyon. And the phenotype happens because the conditions are slightly different on either side of the barrier. So the different environment leads to a different phenotype. So over time it becomes more common by natural selection, because if you have the beneficial allele, you will more likely survive and pass it on to your offspring. So the frequency will differ depending on the conditions and the mutation rate. So there'll be a variation on either side of the barrier. So over time, these two groups will change so much that they won't be able to breed with one another anymore, at which point they will become two separate species. So what evidence do we have for evolution? So we have evidence from molecular biology, from DNA and proteomics. So from DNA, the theory of evolution suggests that all organisms have evolved from a common ancestor. Evolution can cause gradual changes in the base sequence of an organism's DNA. So the more recent a divergence, the more similar DNA will be between the two, because less time has passed for changes in the DNA to occur. For example, if we compare humans, chimps, and mice, we all have evolved from a common ancestor. Humans and chimp DNA match at 94%, but human and mice DNA only at 85%. And this is because we diverged from mice much longer ago. So I have a picture here which shows mammalian forelimbs. So even though all of these have become adapted for different functions, all the forelimbs of all the mammals are constructed from the same basic skeletal elements. So we can see a human, cat, a whale, and a bat. So we can see the large bone in purple is attached to two smaller bones, which are orange and tan, the same here, the same here. And then this is attached to several smaller bones, which are shown in gold, attached to several green bones, and 5 digits in blue. So we can see that there is clearly a relationship between all these. We can also use proteomics, which is the study of proteins, where we look at the size, the shape, and the amino acid sequence as evidence for evolution. So the sequence of amino acids is coded for by the DNA sequence in a gene. So if we're related, there will be similar DNA sequences. So how is this evidence validated? So in a scientific inquiry, scientists first make observations, they then collect the data and use inductive reasoning to draw general conclusions. The scientific community accepts the theory of evolution because it has been discussed and validated, and it's now considered to be valid and reliable. Scientific work generally tends to be accepted once it has been confirmed by the community through either scientific journals, peer review, or conferences. So scientific journals are used for publishing journal articles to share new ideas, theories, experiments, evidence, or conclusions. So for example, the journal Nature and the British Medical Journal also have a policy of archiving data, which allows others to test the data and repeat the experiments. So once the experiment has been replicated and the same results are obtained, it can be confirmed as reliable. The peer review is done before publishing. The scientists can check that the work is valid and supports the conclusion. So again, validating the evidence. And finally, conferences where scientists in a particular field meet for lectures and seminars, and this allows for scientists to discuss and pose questions, which again helps to validate new ideas, theories. Thank you very much. I hope you enjoyed this video.